Hello, everyone, and I'm very happy to come here today to give this seminar. And everything started because we had a meeting of Galician researchers abroad in Brussels, and that's where I met Marta. And so she told me that she's now a Ramon y Cajal Fellow here. And we had this idea that I can come, since sometimes I'm teleworking in Galicia. And so today I want to give an introduction to the type of work I've been doing in Karolinska Institute, and then a few of my latest studies that we are doing now. So I work with neuroimaging and fluid biomarkers with prognostic value in aging and hereditary dementias. I am a senior researcher and a docent in medical imaging. Docent is a title that exists in Sweden, but I don't think it's easy to translate. But it's kind of like an accreditation. And yes, I've been there since 12 years, for the last 12 years. This is the campus in Solna, is the main campus. And we work, this is called Aula Medica. It's a conference center. And in front of it, this is the new, they call it Nya Karolinska. It's a very recent building. It's the largest hospital in Stockholm. And we work next to the hospital in Bioclinicum building on the 10th floor. It's a research and clinical facilities all together in the same building. We also collaborate with other hospitals in the region, in particular with the Karolinska University Hospital in Huddinge. That's in the south of Stockholm. And again, there is a hospital, and next to it there is also a research institute. It's called NEO Building, Campus Flemingsberg. And we also have a long-term collaboration with Uppsala Pet Center. So Uppsala is one of the pioneer centers in the world in developing pet imaging. So basically, I have a background in physics and also in nuclear engineering for my PhD. I did my physics degree here in Santiago. So after I finished my PhD in the US, I had already taken some classes in medical imaging, and I found it more and more interesting to have this medical application. So when I moved to Sweden, I started a postdoc in neuroimaging and focusing on neurodegenerative diseases, especially Alzheimer's disease. And we combine clinical and research data on many types of data, including neuropsychology, in vivo molecular imaging, as well as other imaging techniques, and many fluid biomarkers. And in the last 12 years, we had a lot of changes, like some of the PET techniques were not available, and they are available now, as I will mention in this talk. And when I started 12 years ago, I was focusing mostly on Alzheimer's disease. As you know, this is a growing problem in the society because we live longer and longer, and there is not yet a very effective, I think, therapy, although there are many recent advances. All this, there is a spectrum of neurodegenerative diseases, and they share in common that they eventually lead to dementia, with aging, with enough time. And currently, neurodegenerative diseases are the 6th cause of death after cardiovascular, cancer, or ictus. But this is growing as other diseases have more effective therapies. So there is a faster increase in Alzheimer's and other neurodegenerative diseases as a cause of death. And among all of these dementias, Alzheimer's is the most common. As I said, it's not just Alzheimer's disease. The second most common, especially below 65 years, is frontotemporal degeneration. And in the last few years, I'm focusing part of my research on frontotemporal degeneration as well. And then there is, so I should say that the main characteristics from the phenotypic point of view is that they affect different cognitive functions, where Alzheimer's disease mostly is reflected in deficits in episodic memory. Frontotemporal degeneration affects executive function. And then there is a range of other neurodegenerative diseases that are movement disorders, including Parkinson's disease, which is the most well-known, but there are many others, like dementia with Lewy bodies, corticobasal degeneration, and progressive supranuclear palsy. And when I started 12 years ago, I was working very closely with the Karolinska Hospital in Huddinge in the memory clinic. And I just wanted to show you the type of material, the type of patient data that we collect as we work very closely with the memory clinic. It's quite a large memory clinic with greater than 700 patients per year. And in this chart, we can see in gray colors are individuals, patients that don't get a dementia diagnosis as they come to the clinic, and in colors, they do get a dementia diagnosis. From the dementia diagnosis, which is 1/3 of this total number of patients, the majority will receive an Alzheimer's disease diagnosis. And as you can see, there are other type of dementias, like the frontotemporal dementia, vascular dementia, Lewy bodies. But 2/3 of the patients don't receive the dementia diagnosis to start with. 1/3 approximately receive what is called mild cognitive impairment. So these are individuals that have cognitive impairment that can be measured objectively, but not yet, they are still independent. So what defines dementia is the lack of independent living. And then there is also a little bit less than 1/3 of the individuals that they have a feeling that they are losing some cognitive function, but when you do the cognitive test, they don't have any significant findings, and they are called subjective cognitive impairment. So for us, for research, actually these 2/3 of the patients are very interesting because we are trying to catch patients early so that we can diagnose them very early and try to find what is the cause of their cognitive impairments. And it's a very important group of people to do research and to develop these early biomarkers. And from a biological point of view, what is the common underlying basis of neurodegenerative diseases? And this would be the accumulation of proteins in the brain. And for that reason, they are called proteinopathies. And this is a very simple diagram showing 3 of the most common protein deposits in the neurodegenerative diseases. One is amyloid, tau, and alpha-synuclein. And the most common is tau, actually. There are many neurodegenerative diseases that share this accumulation of tau protein deposits. Now we have PET tracers for both amyloid and tau. And there is a strong interest to develop PET tracers for alpha-synuclein, but it's not yet available. And as I said, when I started, there was only amyloid PET. And just a few years ago, we start to have tau PET tracers. And thanks to this progress in research on biomarkers, now there is a tendency in the field towards a biological definition of neurodegenerative diseases, including Alzheimer's disease, prior to clinical symptoms. And Alzheimer's is defined by the accumulation of both amyloid and tau that we can measure in the brain with PET tracers. In addition, a third component of Alzheimer's disease and other neurodegenerative diseases is neurodegeneration, which is atrophy, brain atrophy, loss of brain cells, but neurodegeneration is not specific to Alzheimer's disease. But we are also working with biomarkers for neurodegeneration. As I said, we work with both structural and molecular imaging. And as you know well, there are advantages and disadvantages, but we combine both structural and molecular imaging. With structural, we get high spatial resolution, and with molecular imaging, we get great sensitivity because we are able to measure with very low concentrations of proteins of interest or glucose metabolism or biological mechanisms. So actually the combination of both gives a lot of information, both high resolution and high sensitivity. These are, in this chart, I want to summarize the PET tracers that we at Karolinska have been working on. Many centers around the world work with amyloid PET tracers. First there was the PIB, carbon-11 PIB, and now there are many others that are labeled with fluorine-18, and fluorine-18 label tracers are more useful for clinics because they have a longer half-life, so you don't need to have a cyclotron in place. Then we also work with FDG PET, which is the most widely used PET tracer because it's also useful for cancer. And recently, tau PET tracers. We started with a tracer from Japan, fluorine 18 THK5317, but now it's not used anymore because it has some limitations, which mostly because it's not specific enough to tau, and it has a lot of, it's called off-target binding. And more recently, we have fluorine 18 fluortaucipir and second generation tau PET tracers such as Merck compound. A characteristic of our center with Professor Norberg is that, compared to other centers, is that she had a very strong interest in imaging biological mechanisms besides proteins, especially glial cells. So other groups did not originally think that glial cells were an important component in neurodegenerative diseases, but a strong interest in our group has been on inflammation. So glial cells, astrocytes, and microglia as key components of why Alzheimer's disease might be caused. And so when I started, I did my first studies using deuterium L-deprenil. It's a tracer that binds to monoamine oxidase B, and it's a molecule that is overexpressed in the brain of Alzheimer's disease patients compared to healthy controls. So I will show a little bit some of those data. And yeah, so these are the types of PET tracers that we work with. Amyloid PET is now used in the clinics. It has clinical utility. And in a very, very brief, the clinical utility of amyloid PET is that if you are positive for amyloid, and you are also mild cognitive impairment patient, you have an increased risk of conversion to Alzheimer's disease in the next 2-3 years. There is an enhanced risk to convert. And if you have a negative amyloid scan, it's used to exclude Alzheimer's disease. And these are a number of amyloid PET tracers that have been approved, and they have been developed by different pharmaceutical companies. But they all measure the same thing, which is the accumulation of plaques in the brain, amyloid plaques. And a very important use of amyloid PET is in clinical trials. As you know now, we are in the beginning of some successful clinical trials of anti-amyloid agents. And there was anti-amyloid antibody called aducanumab, and now it has been retired from use, but because, so when you look at the before and after, so individuals that have been given this therapeutic agent, 1 year later, amyloid PET scans revealed that it's a very dramatic reduction in the burden of amyloid. So it really showed that this pharmacological agent was able to remove amyloid from the brain. However, the therapy is not effective because it's not the patients don't have a good enough clinical outcomes. So they still don't, they keep getting worse with time. So it didn't work, but now there is more trials. The most successful one is called lecanemab. And well, there are questions whether it will be commercialized in Europe. It's not approved yet, but it's approved in the US. So this is a very recent topic, which is clinical trials. And because we know that clinical trials are coming for amyloid PET, in Europe, we have been involved in a large consortium called AMIPA. AMIPA is amyloid imaging to prevent Alzheimer's disease. So before we have the therapies, we want to know as much as possible in a very large cohort of people across Europe. It involves all of these centers. So some of them are in Spain. Karolinska was involved, London, Amsterdam. There are many centers in Europe. The idea is to collect thousands of amyloid PET scans. And some of these cohorts have been enriched by recruiting participants with affected family members. As you know, part of the cause of Alzheimer's is genetic. So if you recruit individuals, like in Barcelona Beta, for example, you try to recruit people who have family history. So you are going to have a more enriched cohort of participants that are cognitively healthy but at higher risk to develop Alzheimer's disease. And these cohorts tend to have, they have an overrepresentation in certain genetic risk factors, such as APOE, that you may have heard of. Is the most common genetic risk factor for sporadic Alzheimer's disease. And there are different versions of the APOE that the highest risk factor is when you have 2 alleles of the APOE4. So you can be 3-3, 3-4, and 4-4. If you are 3-3, you are neutral. You don't have enhanced risk. If you have one of the alleles, your risk increases. And if you have 2 of them, your risk increases even higher, even more. But it's not deterministic. It doesn't mean you will develop Alzheimer's disease, but it increases your risk. And I would like to also point out here, before I go to some of the recent results from our group, is that we are collecting data on amyloid in the brain using PET imaging, but we also collect cerebrospinal fluid and plasma, because we can also measure amyloid in the fluids, right? We can take lumbar punctures. These are samples from the spinal cord, and blood tests. We have data on all of these 3, the brain, spinal cord, and plasma. The amount of data from these different type of data depend on the country. For example, in Sweden, people are very open to get lumbar punctures. It's very common. Also, some research in other countries, like in the US, is much less common because it's considered intrusive. You always have a small risk to have some problem. It's not a fun procedure. I don't know how it's in Spain.
In Spain, it's very common.
It's very common. Okay, so I didn't know so much, but I know that in Sweden is like almost nobody says no to the lumbar puncture. So there is a high trust also in the system and in the clinics and everything. So, but in the US it's not. People prefer to get a PET scan than a lumbar puncture. Okay, so and before we go to the rest of the results, I should say that having a lot of amyloid in the brain, so amyloid positive in the brain, can be measured by the opposite trend, which is reduction in amyloid species in the cerebrospinal fluid. So it's like they have opposite values. But having high amyloid in the brain is related to having low amyloid species in the CSF. And this is believed to be that as amyloid accumulates in the brain, it gets clear from the liquid. So this has been well known for a long time. So as part of AMIPAD in our group, I have been supervising a PhD student, and we decided to investigate further this idea of measuring amyloid in the brain or in the CSF. So generally, other researchers, other groups in general, we think that there is a high concordance. So you can measure amyloid in the brain or in the CSF, and typically you are positive both in the brain and in the CSF, or negative both in the brain and in the CSF. But the more that we look at data, we realize that there is quite a bit of discordance. Even up to 20% of individuals have contradicting results. So if you do a PET scan, you think you are negative, but if you look in the CSF, you are positive, or the other way. And this amount of discordance is especially high at early stages, when you are not yet MCI, when you are not yet cognitively impaired. It's called early stages of preclinical Alzheimer's disease. So we decided to take data from ADNI, which is 800, more than 800 individuals. ADNI is a data set that is shared among internationally. So it's very, there are many hundreds of studies from ADNI because it's a good way to pool data together. So, and that's why I also thought big data and machine learning and all of this is a very, is for the future, because we are collecting more and more data that is openly available, that we don't need anymore to recruit our own patients all the time. So we can work basically with free data, which is now more and more the tendency. Okay, so we decided to study these two pathways, and we found that by looking longitudinally, as we had repeated measures 2-year follow-up in a cohort of more than 800 individuals, we were able to find the two pathways. So some people first become positive in the CSF, and some people become first in the PET. And we did a very thorough statistical analysis to find if there are differences between these two pathways, and indeed, so we found that one of the pathways has different genetic factors. So looking at the ones that first are positive in the CSF had lower frequency of ApoE44 and a slower accumulation of amyloid in the brain. So this study is one of the studies that shows that getting Alzheimer's disease doesn't mean that we all, so it's very heterogeneous, because there's a combination of genetic factors like ApoE, but also lifestyle and gender. So there are, the more that we do studies on Alzheimer's disease and other neurodegenerative diseases, we realize that they are heterogeneous pathways. And that's the point of our research. So a continuation from this study that we just got accepted in neurology, and it will be published very, very soon in the next few weeks, is that we continue collaborating with AMIPAD, and we had, as you can see here on these graphs, we look at every individual point. Every patient or individual had both amyloid PET and CSF, amyloid in the CSF. The more amyloid in the brain, the lower the amount of amyloid in the CSF. And we develop a continuous measure of the discordance between these two. So if you are concordant, you will be in this hyperbolic, let's say, function, but we develop a continuous index that the more further away you are towards greater amyloid PET compared to CSF, then you have a higher aggregation score. We call it A-beta aggregation score. And it's a continuous measure. So we have also used a discovery cohort from ADNI that we also validated it in, so the validation graph is not here, but we have validated it in another cohort from AMIPAD. So these are European data. So one of the ideas also in many research studies is to have two independent cohorts to try to have more robust findings. So after we developed this continuous measure of aggregation, we also wanted to investigate if there are genetic differences. And we found again, so this is a confirmation from the previous study, that having 2 copies of ApoE4 is related to this aggregation score. And we found this that replicated both in the discovery cohort from ADNI and in the validation cohort from AMIPAD. Okay, so I would like to, was to show you this, our biomarker studies in the brain and in fluid biomarkers. And now I would like to show our studies, just to show you a little bit what we are doing with tau PET and tau biomarkers. So actually, tau protein is a very interesting protein because it's, in normal function, it contributes to the stability of the neuron, of the microtubules, but then it gets hyperphosphorylated, and the biological reasons are not fully understood, but it leads to the degradation of the neuron and the accumulation of the tau proteins, which are degraded into neurofibrillary tangles. These are some examples of tau, how it accumulates, how we can see it in the neuropathologically in autopsy brain tissue. Tau is known to start to deposit in the hippocampus and entorhinal cortex and to spread laterally towards other lateral temporal regions and eventually towards most of the cortex. And these are examples of tau PET scans that you can see that they are highly spatially correlated with what we see in autopsy brain tissue. And as I said, these are examples of first generation tau PET from a review paper from our group and second generation tau PET tracers. We started with THK tracer from Japan, but it has high non-specific binding. So we now work with second generation tau PET. As you can see from the color scheme, there is much more clear distinction between high tau regions and low tau regions. So it's a more sensitive way to measure tau. And yeah, these are examples from a second generation tau PET tracer. It's called Merck 6240. So it has a very nice separation between cognitively normal individuals and Alzheimer's disease patients. And as I said in the title of the talk, I'm very interested in prognostic value of biomarkers. And this is a study from the US. But I wanted to show you that there is an accumulation of studies showing that the combination of both amyloid and tau lead to cognitive decline. And in this graph, they have separated a cohort of cognitively normal individuals into 4 groups, depending whether they are amyloid negative, tau negative, amyloid negative, tau positive, amyloid positive, tau negative, and amyloid positive, tau positive. Amyloid positive, tau positive is the red line, which you can see a clear, faster rate of cognitive decline over time. So the combination of amyloid and tau are very high predictors of future cognitive decline. So now I wanted to show that that's why we have a very strong interest in tau. And recently in January, we had our student, Mona Lisa Mallard. She's from France. She just graduated and got her PhD. And the focus of her research was tau. And part of her thesis was on in vitro autoradiography studies with tau PET tracers, and one of the studies that I was helping to supervise, she worked on in vivo tau PET. And I want to show you a bit because I thought in this study we wanted to not only focus on amyloid and tau, but in additional biological mechanisms that are influencing cognitive decline, and we wanted to investigate, in particular, peripheral inflammation. So, as I said earlier, one focus of our group is inflammation in the brain, which is mediated by astrocytes and by microglia, but there is much less known about can we measure inflammation in the blood, and what does it mean, right? Is the inflammation measured in the blood related to changes in the brain? So we wanted to investigate the association between amyloid in the brain, inflammatory markers in the plasma measured with Olink inflammation panel, and tau deposition in the brain with a second generation tau PET tracer. And we had also many other markers of neurodegeneration, cognition, and we selected a cohort from the US. We requested this data. Again, this is an example of sharing data, because we didn't have data on second generation tau PET. These are very expensive studies, but we requested it through these databases. You have to make a request for a specific topic, and we got data on 132 cognitively normal Hispanic individuals from New York, from Manhattan, that had many biomarkers. So these are some of the examples of biomarkers: amyloid PET, tau PET, T1 weighted magnetic resonance imaging, and FLAIR magnetic resonance imaging. We had a marker in the blood called neurofilament light, which is considered a surrogate marker of neuroaxonal degeneration. So this is a very now popular marker, neurofilament light, because it can be measured in the CSF and in the blood. And it is believed to be strongly correlated to especially to the rate of neurodegeneration, how fast we are having atrophy in the brain. So we had data on this neurofilament light, and sorry, it's a bit busy, this slide, but we divided our 132 sample of cognitively normal Hispanic individuals into 4 groups, depending on the levels of amyloid and neurofilaments in the blood. We wanted to investigate plasma inflammation, amyloid and tau in all of these different groups. Okay, so first, as you can see, we have the group 0, 1, 2, and 3. These are groups depending on the amyloid and neurofilament light. So we found that first we look at the amount of tau in these different groups. And we found that to have tau in the brain, you need to have amyloid. So group 2 has high amyloid, and then they also had high tau. while the other groups did not have such a substantial amount of tau in the brain. Good, but we wanted to investigate this group 3 because the group 3 has low amyloid but has high neurodegeneration. And not so much is known about this type of individuals because it's not the typical Alzheimer pathway. Alzheimer is characterized by amyloid in the brain. But they had neurodegeneration because we could measure high plasma NFL. And what we found is that this group has atrophy and also has a measure of cardiovascular injury, which is white matter hyperintensities. But most interesting, and I think this was a new result, is that we found high amount of plasma inflammatory markers only in this group 3. So group 3 has high neurofilament lights, which is ongoing neurodegeneration in the brain, and they also had high plasma inflammation. And these are the proteins that were most strongly correlated to neurodegeneration in the brain, and especially one of these plasma inflammation markers, which also we found that this cohort is enriched in many factors that are called comorbidities, so it's body mass index and diabetes. So in summary, finally, they also had low episodic memory, and only in group 3. But we want to summarize these findings that although most of the studies focus on amyloid and tau, and this is the most typical way to get Alzheimer's type of cognitive decline, we also found a comorbidity driven with chronic inflammation that also leads to neuronal injury. So our study suggests that we should be more open-minded when we look at the development of Alzheimer's disease, that is not all about amyloid and tau, but there is a high importance of considering alternative biological mechanisms that contribute to cognitive decline. And some of them are inflammation or cardiovascular injury, diabetes, and body mass and obesity. Okay, and then in the final type of technique that we use, I would like to point out that we also want to take advantage of structural MRI and diffusion-weighted imaging. And here we are looking at diffusion-weighted imaging in the cortex, in the gray matter. So traditionally, diffusion-weighted imaging has been mostly evaluated in the white matter tracts, but in collaboration with Hospital Sant Pau in Barcelona, with Juan Fortea and Victor Montal, we are now using this technique to investigate early structural changes that are called microstructural changes. And overall, the idea of this technique is that we think it's more sensitive than structural atrophy measured with magnetic resonance imaging. So it's just a way to try to get a little bit early microstructural damage. We are performing multimodal integration of structural MRI with DWI. So that way we get in the same study macrostructural and microstructural changes. And we use different software techniques, in particular FreeSurfer, to achieve the accurate surface-based registration of these two types of images. We have different corrections, like partial volume correction and smoothing on the surface to minimize contamination from white matter and the CSF. Okay, I'll go a little bit quicker with this. Is just to show more details about the different software that we use. FSL is to perform co-registration and skull stripping and the DTI fitting of the tensors. And then FreeSurfer to perform the statistical analysis at the surface level. Our type of analysis we can do on the surface is called vertex-wise analysis, or we can apply atlases and extract ROI-based, it's called regions of interest. So these are just different ways to look at the data. We have, the first time in our group in Karolinska, we have applied this technique. It has been in a cohort from Harvard Aging Brain of older adults. And the main, I will also not spend too much time. This was published in 2021, I think. But the main idea, and I want to show the motivation. So as I said, both amyloid and tau predict, there are markers for prognosis that individuals will decline cognitively. But if you only have amyloid pathology, so if you have amyloid PET, this is a risk factor to predict cognitive decline over the long term, maybe like 6 years later or a long time. So what we wanted is, for clinical trials, usually you don't have 6 years or 10 years. You want to have results in 2 or 3 years. So we want complementary and non-invasive imaging biomarkers to help us to identify those individuals at risk of short-term cognitive decline. And the hypothesis was that by combining amyloid and diffusion-weighted imaging, we might be able to find those individuals that will cognitively decline in a short time, such as 2-3 years. And therefore, it could be a useful predictor of individuals at risk for cognitive decline, and therefore that they are the best to be selected for clinical trials. And basically, diffusion-weighted imaging measures movement of water molecules. So when you have high microstructural damage, you can detect an increase in cortical mean diffusivity. So in this cohort from Harvard Aging Brain, we had about 196 participants. And it's a very rich data set because it's baseline data, but they also collect not only diffusion-weighted imaging, but also magnetic resonance, amyloid PET, and tau PET. And also they have cognitive assessments over several data points in time, because it's an ongoing study. They keep collecting. When you have, all of them are cognitively normal at baseline, and when you have a cognitively normal cohort, about 25% are already amyloid positive at baseline. Okay, so their main result is that we found that we wanted to see if cortical mean diffusivity is more related with amyloid or with tau, and the main result we found is that we did not find an association with amyloid, but a cross-sectional association with tau. So, and then since I said that we are interested in biomarkers that have prognostic ability, we also found that cortical microstructure can predict cognitive decline, and we ran many different multivariable linear regression models. The main finding that I want to summarize here is that cortical mean diffusivity is able to predict this cognitive decline even after correcting for cortical thickness. So we think that cortical mean diffusivity is a prognostic biomarker that adds value over measures of cortical thickness. So microstructural imaging has independent information that has prognostic value. And besides predicting cognitive decline, it also predicts the rate of hippocampal volume loss, and it also predicts the conversion from cognitively normal to mild cognitive impairment. And in a more recent study, we also show that it predicts accumulation of tau over time. And then I think I need like 10 more minutes. I want to introduce those. So this is the group I am working together with. There is a doctor called Caroline Graf. She's a geneticist, but she works part-time research and in the clinic. So she sees patients. She's trying to find people with mutations. But the mutations that she's trying to find are called autosomal dominant. And these are, they predict with, so if you have the mutation, you develop the disease with 100% probability. And in Sweden, we work with Swedish familial Alzheimer's disease. These are studies on autosomal dominant disease that has been collected since the 90s in a longitudinal design. And we have data for 81 individuals and in a total of 219 visits. In this group of individuals with familial Alzheimer's disease, we found that cortical microstructure is correlated with astrocytosis. And we can see the pattern of increasing cortical mean diffusivity from presymptomatic to symptomatic stages of autosomal dominant Alzheimer's disease. And then more recently, we are also collecting data with many plasma biomarkers, including total tau, phosphorylated tau, neurofilament light, and a marker of astrocytosis, which is GFAP. We published recently that the earliest changes occur in glial fibrillary acidic protein, GFAP, that starts to change about 10 years before clinical onset, followed by phosphorylated tau, and finally NFL, which occurs more at estimated years at disease onset, at clinical onset. Similar findings have been found in a larger cohort from the international DIAN studies. So it's similar to our findings. And also this year we combined data from PET and GFAP. And not so much is known about GFAP as a marker of astrocytosis in the plasma and deprenyl in the brain. And in this paper, we have reported that they actually follow opposite patterns, where GFAP increases with time, while deprenyl is highest in the earliest presymptomatic stages and declines with estimated years to symptom onset. And finally, I want to have a couple of slides to show you our studies in familial forms of frontotemporal degeneration. This is a very heterogeneous type of disease. It involves the accumulation of different types of proteins, not only tau, but also TDP43. And we work mostly with progranulin mutations, C9orf, and as well with mapped mutations. Since frontotemporal dementia is quite rare, we collaborate with GEMFI, international studies. We have non-carriers, presymptomatic carriers, and symptomatic mutation carriers. And a lot is known about atrophy patterns, and from accumulation of studies from international GEMFI, and they report that there are differences according to the mutation type. So there are different patterns of what brain regions are affected, where mapped mutations that are characterized by tau affect mostly the temporal regions, and C9orf and progranulin are more affecting frontal regions, and very widespread patterns in C9orf. And we hypothesize that our measure of cortical microstructure can track early neuronal injury with more sensitivity than cortical thickness. And these are ongoing studies. We combine with CSF data. We found some of the key proteins to be characteristic of frontotemporal dementia. And these are neurofilament markers that are markers of neurodegeneration, but also other markers of glial cells, such as aquaporin 4 and BGF. And there is an ongoing study that is unpublished, combining MRI and CSF. And we are working with cortical thickness, comparing with cortical mean diffusivity in the different mutation types. And finally, we are already looking at longitudinal data, and we hypothesize that the longitudinal rate of increase in cortical mean diffusivity will increase from non-carriers towards presymptomatic carriers and symptomatic carriers. And we want to compare with longitudinal rates of GFAP and NFL. So this is the last slide. In summary, we want to find biomarkers that are characteristic of different stages, from preclinical to MCI and to dementia. And we think that our studies contribute to finding the timeline of neurodegenerative diseases, so using both PET tracers, structural, microstructure, and all of the range of plasma and CSF fluid biomarkers. Thank you very much. And these are some of our international collaborators and the funding agents from Sweden and European funders. Thank you. You have some questions? Or?
First, I wonder, because I'm not very familiar with PET imaging, how reproducible is that? Does it change a lot?
Yes, test retest. Yeah. No, so actually, no. I think it's very robust. And before using a tracer, before widespread use of a tracer, it's called test retest studies. So you need to check that if you take the same PET scan the next days, or that the variability is not more than 5%, for example. Yes. And also because the way we analyze data, we try to use the same atlases and the same brain regions, so we can compare across centers. And of course, there is a lot of, if you are going to use it in clinical studies, there is a lot of effort to harmonize across scanners, also the methodologies, to try to use similar methodologies. Yeah.
And I also have two curiosities, because I was working with the ADNI dataset a long time ago.
Yes.
So is this still the same dataset, or are they quite new scans from
It's always growing, this data. Yes, yes, yes.
About launch a new branch, actually. ACNI4 is about to start next year.
Yeah.
So it's ACNI1 was focused mainly in MRI data
Yeah.
and FTG PET, and it was composed mainly in cognitive normal subjects. And then ACNI2 and ACNI-GO that were the following projects. So the finding is coming like every 5 years. This was mainly focused in amyloid PET and ACNI3 in tau PET. And now is ACNI4 is about to launch.
Yep.
How many patients are there?
Thousands. Yeah, I mean, like you have to search what is your research question. So when we checked, we wanted people who had at least 2 follow-ups with CSF, amyloid, and PET, and we found 800. If you look for tau PET, there was not so much, but now it's growing, I think. I don't know if there is already second generation tau PET or not yet. Maybe not.
Yes,
Fluor tau CPIR, yes, because I think it will be later. And that's, yeah.
there are like 2,000 scans, I think, but they are not from 2,000 different patients. Some of them are manipulated.
Yes. And that's why we wanted with the student, we needed to do second generation tau PET, so we could not find in ADNI, and we had to contact other groups. But there are ways to get data. Also, there are these A4 trials with Risa Sperling, and they want to share. Harvard Aging Brain is now sharing as well. So the tendency is to share, because I think they realize that after they have published some papers, it's much more worth to share. Because there is no, I think at the end, the limitation is people to work with the data. It's time consuming, and you need the students and you need postdocs. I believe that before, maybe 10 years ago, it was, for example, Agneta that I work with, the limitation is money, because these scans are very expensive. There are thousands of euros just one scan, but now things have changed. Now we work in different ways. We share data. So I think it's now to find people and time to analyze, I think.
One very good thing about the neuro communities, like they also share the post-process data, as well as I remember.
Yes.
Because, for example, with cardio,
they share. Yeah. They do. You can get raw data or you can get process data. There are different options. Sometimes they also share the Excel data, so you don't need to process again. And they do this in Harvard Aging Brain. They're sharing the Excel sheets. They also, if you ask, they can also share images. But I mean, the easiest way for them is to share the Excel files, because you can download.
Actually, several different processing pipelines from each of the.
Yeah, actually, these days you can do a lot of research just with Excel data, don't you think? You don't need to see an image. You're doing neuroimaging and you don't see an image. No.
We still need to work.
Of course, it's a lot of work. I mean, I'm not saying it's not work. You know how it is. I'm not saying it doesn't have merit, but yeah.
And the CT, you don't really use CT?
No.
Because one image in the beginning, but
No, I don't use CT. No, because I don't think you get enough. I don't think you get enough information for research. Yes. Do you have a question, Pablo?
Do you have some experience using kinetic modeling in tau PET? Because people are working with static PET images.
Yes.
And I don't know if there are added value.
Yes, we did in the group. Konstantinos Kiotis from our group did his whole PhD on tau PET. It's the first tau analysis in Sweden, and he, in collaboration with Uppsala, we did PMOD. We use PMOD to do kinetic modeling. It's a lot of work because, yes, I'm more into, I don't find it so fun that you focus on these very early pre-processing steps in kinetic modeling because it's a whole research topic on itself. So I haven't focused so much, but I have done it. I have done for the Prenil as well. We use PMOD. We collaborate sometimes with Uppsala, and there you have like Mark Lubering, so they run models for us. But I think if you want to do that, it's almost like you have to only focus on this, because it's a big, don't you think so? It's a big topic to optimize the model. Yeah.
The progress of the tau tracers.
Yes.
There are a
Yeah.
lot of
Of target binding. Yeah.
I don't know. But maybe we have not enough data.
Yes, we had a very small cohort, because actually very small, 20 patients in total. Maybe it was 9 MCI, 11 AD or something like this. But I think it was closely correlated with SUVRs. So I don't think it was a huge advantage for tau PET. But sorry? Or for any tracer, for that. Maybe the Prenil was sensitive, because it's very, but why? Because the Prenil is not totally reversible tracer. So it's needed. We need to model it. It's not the best. At least the one we use, the carbon 11 Deprenil, had. If you look at the time activity curves, they were not like nice. This like the flash out was not like that. It was more like this. It tends to stick a bit. So it's in between reversible and irreversible. It's very painful to model and to yes. And also because there is no good reference region, so the cerebellum is not totally free from MAUI, and so it's a mess, because when you don't have a good reference region, it's really hard to... The kinetic modeling, we call it pseudo-reference region. We still use the cerebellum, so yeah, I think it's difficult. Are you trying to do kinetic modeling or tau, or yes?
Doesn't make it depends on the tracers, I think. In proteinopathies, it doesn't make a lot of sense.
Yeah.
Actually, because the tracers are fully reversible.
Yeah.
SUVR is a very good approach.
Yes, totally. Yes, exactly. Unless if it is a very new tracer, you still don't know the behavior, but yeah.
Another question is about the value for the future role of tau PET in the clinic. Do you think that there are added value about the regional distribution of the tracer compared to
just like a composite or meta ROI or something?
Yeah.
You mean is enough with?
Compared to blood biomarkers, it's
Yeah.
very expensive.
Yeah. Okay, I think you need added value. Otherwise, people are going to use p-tau. Now we have very good plasma markers of phospho-tau. So I agree with you. In order for tau PET to have clinical value, it has to be used all the topography because that's the value of tau PET, right? If you are just going to use one number as a composite, then you get the number from the blood.
Exactly, but we don't know if this is interesting for the clinic.
No, not yet. We don't know because it's also the cost. It's more expensive. So that's a huge, yeah. No, I agree, but it has to have a lot of added value from the topography of tau to be able to justify the cost. That's what I think.
There are some interesting papers, one from Jake Vogel, that he does this sustained methodology.
Yes.
He finds different subtypes that have different prognostic values.
Exactly. But I think for research, it's so interesting, tau PET, and all the topography, right? I mean, the clinic is another story, but we learn so much from the heterogeneity of the disease.
Maybe for the follow-up.
Yes, and also in the future, the frontotemporal dementia, we still don't have good tracers. We don't know where the proteins are deposited in vivo. This is a huge field for research whenever we have any tracer. For TDP43, for 4R tau, right? So whenever there is any of those tracers, we have a huge, very interesting research to do. because now we don't know. We only have autopsy data. So yeah, in our group, we also have a biobank. So we want to later compare this microstructure with some autopsy data. But that's all we can do, because we don't have, we can only look at the end. Yeah.
I have also a question. It's related, though a different topic probably, that is diffusion MRI.
Yes.
And in your group and also in the group of San Pau,
Yes.
you focus a lot on mean diffusivity
Yes.
as a measure of microstructure.
Yes.
But there are a lot of different indexes that you can extract
Yes.
from
Yeah.
mean diffusion. From
Yes.
diffusion
Yes.
data.
Yes. Yes.
And why, for example, fractional anisotropy, for
Yes.
example, by principle, it wouldn't make a lot of sense
No.
to measure it in the cortex.
Yes.
But there are a lot of other options.
Like
have you explored?
Yes. So I have not, but like free water and other. Yes.
For example, there are.
Yeah, I haven't. Because for me, we don't have a lot of people right now, and so with Caroline Graf, I am the only one doing it. So I want to start with to finish first the mean diffusivity to see already what we get compared to cortical thickness. But we will have all the data in the server and ready to look at other properties. And I know that Victor, for example, has published in Alzheimer's and dementia where the reviewers ask for this extra information. So I think it's in the supplement, or at least in answering the reviewers. He has done this. And I think our script allows to get these numbers. Yeah.
There are some interesting papers working in the mode, for example, that would be very easy to extract from the tensors.
Yeah. Yes. I'm very interested, and I'm also in contact with other people in San Pau that also part of GEMFI, and we want to do complementary studies on the same set. So we don't want to reanalyze everything. We want to share the process images and look at more things. So if you have ideas, we are very happy to collaborate, and with San Pau as well. I mean, we are collaborating with San Pau now, so because they want to also look in the subcortical areas that we are not looking. And then we want to combine it all to have more information. Yes?
Thanks for the talk, because it was very, very interesting to see how we could complement to what we do here in CiTIUS, because we have here several research groups in medical imaging, in prediction models, and so on. You mentioned that there is a role.
Yes.
There is always a role for AI almost everywhere.
Yeah.
I see from your presentation that you are not working really on using AI or prediction models or machine learning.
No, I am not. No.
Not specifically about. What's your vision about the role that, for example, machine learning or prediction models or medical imaging could play in this?
I think a huge role, because especially since this year, I think is coming. Everyone is interested in doing AI for everything. And at Karolinska we have this SciLife lab. They do big data. It's growing a lot, and they are applying AI. And there is a lot of workshops all the time. AI for medical imaging, so it's coming, and people are very interested. So it's a very hot topic. So there is a huge room. Me, myself, I haven't, because I didn't get yet. I'm busy with processing the images, and so, but I'm very interested to collaborate on machine learning and AI. Yes, I don't know what you think, but and I'm also like evaluating grants, like commission, and now it's almost every grant has AI in everything.
They say they have AI, but
or they want to do.
Not all cases with background and real knowledge about what to do, what's better to test or not. So I think that would be interesting to work from both sides, because there are certain people here.
I think we should explore doing common analysis, and I'm definitely. Yes, but I'm very ignorant about AI, and especially all these questions now, the trustworthy AI and robust system, and is changing so quickly that it's really hard to keep up. For example, now evaluating grants, they ask every evaluator, please evaluate the robustness of the AI method, evaluate the trustworthiness, the ethics, and also the environmental. They're asking things we cannot do. I mean, I'm scared to, no? How do you do this? Isn't it,
It's not easy.
nobody knows.
No, of course, many people knows, but probably they are not asking that to the right people.
No.
They should have an expert on this part of the
Yeah.
topic, because
Yeah.
it's not straightforward to assess
Yes.
worthiness or
No.
robustness, even for AI specialists.
Even for AI specialists. So I've been in panels that they try to have at least one person who is a specialist in AI.
Yeah, but
So-
this doesn't mean that this person is also a specialist on trustworthiness.
No.
They may be interested on
Yeah.
other
But
metrics.
I have the feeling that the trustworthiness topic is something of this year. I never heard of this. Or have you been talking about it for some time?
Yeah, at least for the last 8 to 10
Okay,
years it's
okay.
been going around.
Yeah.
And now it's been increasing a lot because
Yeah.
of the AI Act, because
Yes.
of medical applications
Yes.
are these critical and
Yeah.
these,
Yeah.
dangerous areas, so they need to look in detail to that.
Yes. Yes.
But
Yes.
This is also a very nice
Mhm.
aspect to consider, because it will
Yes.
be
We should.
necessary.
I think maybe even starting looking at grants, like consortia, apply together and explore. No?
It's very
Yes.
interesting to see
Yes.
both sides of the
Yeah.
the clinical and the medical research area, and imagine how we
Yes.
could
Yeah. And the doctors really need these engineers because they cannot do everything. So they don't have time, and they have accumulation of data. So yes. And we have a big biobank with plasma samples from Stockholm, not only from Genfi, but we have a database that we spend every week work on the database. So because it's continuously growing, and donating brain, not only genetic forms, but sporadic forms. So my supervisor, Karolina, is the head of the brain bank. So she manages all the brain donations from in Stockholm, at least. So it's also
And
good.
And in Spain, you work with Barcelona.
Yeah, for the moment, yes, I work with San Pau.
And before you say something about Barcelona or something,
Mhm. Barcelona Beta. Yeah,
that's the journal already.
yeah. Because it was part of AMIPAD, and they focus a lot on amyloid, I think. Well, many things.
They now are more focused in fluid biomarkers, I think.
Yes, that's true. Plasma.
Mark is there.
Yeah.
But they had the alpha study that was one
Yeah.
of the first amyloid studies.
Yes. And now it's Alpha Plus also. Yeah.
So we have more questions. We thank you again.
Thank you.